In this module, we will discuss what is the cascade of the RAS map kinase. We have mentioned it times and again that the RAS map and the controlling factor, it acts in a cascade manner. And how it is there for controlling and for dealing the cascade mechanism. Now, RAS GTP, as we know, is an interact directly with several downstream targets. जो कि बहुत से downstream targets हैं, उनके साथ इसका क्या है? It interacts. And the cascade gives a response to a wide variety of extracellular जो signals हैं, and it plays a key role in the regulating vital activities. Key role vital activities कौन-कौन सी हैं? Cell proliferation, कि cell की growth, and the differentiation. Remember, the cell proliferation को control करना बड़ा ज़रूरी है. Because if you won't control the cell proliferation, it will become oncogene, it will become a cancer. Similarly, there ought to be a differentiation. Cell की differentiation की भी बड़ी ज़रूरत है कि इसने कहाँ कहाँ जाना है, किधर किधर इसके signaling है. Now, for example, this is the step of generalized map of kinase जो है उसका cascade है कि किस तरह ये RAS from top to bottom it travels and it attach with different proteins, with different molecules, and from one stage to another, and eventually in the top, in the bottom, it produces the effect, the signaling effect. Now there are GEF for RAS, that are many activated receptor tyrosine kinases. They possess phosphorylated tyrosine residues, and those residues act as the docking site for the adapter protein. Remember, we have already discussed, and we know that there are docking proteins, there are adapter proteins, and so forth. So GRB2, that is in turn bind to the SOS, which is a guanine nucleotide exchange factor, which is known as GEF for RAS gene. Now, GRB2 binding sites, they create GRB2 binding site on an activated receptor, and it promotes the translocation of GRB2. ये जो GRB2 की translocation है, उसको promote करता है, and SOS from cytoplasm to the cytoplasmic surface of the plasma membrane. अब देखें कि cascade कैसे बन रहा है कि it is going from cytoplasmic membrane, from cytoplasm to the cytoplasmic surface, and then to the plasma membrane of the cell where it is going. Just like this, you can see the active RAS is there, ligand bound compounds. These are the adapter proteins, GRB2, and it functions as a link between other proteins. GRB2 and the ligand protein, they are joined with SOS, and at the end, there is RAS GTP molecule, which is the active form, and it is bound to the plasma membrane. Cytoplasm say cytoplasmic membrane, and then to the plasma membrane, it has taken them. RAF is a serine threonine protein kinase. RAF क्या है? इसका ये एक serine threonine protein kinase है, and one of the substrate is the protein kinase MEK, जो कि MEK, which is activated as a consequence of phosphorylation by RAF, and it goes on phosphorylating and activate two MAP genes which are known as ERK1 and ERK2, जिनको ये phosphorylate करता है during the process. Now there are 160, more than 160 proteins that can be phosphorylated by these kinases, जो कि इन kinases से फॉस्फोराइड हो सकते हैं एंड दे हैव बीन आइडेंटिफाइड. वो कैसे उनकी शनाख्त है, कैसे वो हैं? दे आर ट्रांसक्रिप्शन फैक्टर, प्रोटीन काइनेजेस, cytoselector proteins, apoptosis regulator, receptors, and other signal protein. So ये 5 जो हैं ग्रुप में, ये काइनेजेस ये आइडेंटिफाई हो चुकी हैं और हैं. Once activated, the MAP kinase is able to move into the nucleus. सेल के न्यूक्लियस में ये मूव हो जाती है, वेयर इट फॉस्फोराइलेट्स अगेन, ये फॉस्फोराइलेट्स करती है विद एटीपी एंड एक्टिवेट्स ए नंबर ऑफ ट्रांसक्रिप्शन फैक्टर. ट्रांसक्रिप्टर फैक्टर जो कि प्रोटीन को बनाते हैं, जो ट्रांसक्राइब करते हैं, उनको ये एक्टिवेट करती है. और दूसरी न्यूक्लियर प्रोटीन जो कि असेंशियल है, जो कि ज़रूरी हैं फॉर द सेल एक्टिविटी एंड सेल सिग्नलिंग. अगेन हेयर we can see the combination between various cyclins and cyclin-dependent kinases at different stages of the mammalian cell cycle. Remember, mammalian cell cycle has got different stages. तो उसमें ये कहाँ ये एक्ट करते हैं. For example, like you can see the cyclin DS जो है, ये जब होता है तो cyclin BA and CDK वहाँ पे ग्रोथ होती है. फिर दूसरी अंदर G1 जो स्पेस है वहाँ पे ग्रोथ होती है. एंड S जो है उस एरिया में जो कि इधर ग्रीन है, that shows the how the different phases of the cell, mammalian cell cycle, they help. Now eventually the pathway which leads to the activation of gene involved in the cell proliferation, जो कि सेल की ग्रोथ में, सेल के कंट्रोल में जो जीन इन्वॉल्व होते हैं, दे इंक्लूड साइक्लिन D1 विच प्लेज ए की रोल इन ड्राइविंग द सेल फ्रॉम G1 फेज टू S फेज, जो कि उसकी ग्रोथ की डिफरेंट स्टेज है. और उसमें कौन सा है इन्वॉल्व? दैट द जीन that is the cyclin D1 which plays a key role in driving a cell from G1 phase to the S phase of the mammalian cell.